So we'll do a quick look over nerves, hormones, and homeostasis. That's all of chapter 6.5. And here are your objectives for the unit. Take a second, read through those, make sure you understand at least what is being asked for. If we kind of break this up, this whole top section here is all nervous system. We scroll down a little bit, and then we move into homeostasis and then diabetes. So here's a diagram of a typical motor neuron. And in looking at a motor neuron, there's a couple pieces that we can take a look at. Up on the end, we've got the cell body. Coming off of that, several dendrites. Each dendrite is going to be connected to another neuron. And so this would be the presynaptic and the postsynaptic neuron here. The signal is going to be sent in this direction from the cell body downwards towards the terminal ends, and these terminal ends synapse at either a muscle cell, as shown here, or it can be a gland. So here's a much more simplified drawing. If you want to go ahead and hit pause now, you should be able to label each of the areas. So here everything is labeled for you. Again, our cell body, the axon, terminal end. If we look at the axon, a couple pieces that we need to make note of. Each one of these are different cells that wrap themselves around the axon. This is your myelin sheath, and these in particular are called Schwann cells. And in between the myelin sheath, in areas where the axon is exposed, those are called the nodes of Ranvier. So here, here, and here, they're shown that you can have millions of Schwann cells going along the axon. So we can go over a quick review of the organization of the nervous system. We've got two parts, your central nervous system and your peripheral nervous system. Again, we can look into the sensory and motor divisions. And within your motor division, more specifically, we can start to look at those actions that you mean to do. And those are your voluntary actions. And then we can also look at autonomic. And within autonomic, you've got your parasympathetic, that's your rest and digest, and your sympathetic nervous system. So if we start to take a quick little zoom out here, it's important to remember that you're always, not always, but you're typically going to have a nervous system impulse that goes from a sensory, sorry, I'm trying to highlight here. Okay. That your impulses are going to start with a sensory nerve impulse that then gets relayed or sent to the central nervous system for some kind of decision making, which is going to synapse with a motor neuron. And so that's going to lead us into the next idea here. And again, here is an example of an action potential. The parts that you need to know, I'll label very briefly, but you can go back to my other video to check that out. So here is your resting potential down here. Once you hit the threshold, you're going to quickly depolarize. And then repolarization happens, followed by a period down here. And this is your hyperpolarization, where the sodium and potassium have briefly switched, and your resting potential is reestablished when your sodium potassium pumps start pumping everything back to the correct side. But again, if you're confused about that, check out my other video. I'll also post a couple other links for you there. And so here you've got the quick schematic of what's going on. Here's the inside of your axon. Whoops. Grab a pen. Here we go. Inside of your axon is here. Here's the outside of your axon. And this is a cell that is in resting potential, meaning that your sodium-potassium pump is working to pull 2 potassium in for every 3 sodiums that get pumped out, leaving you with an overall negative inside, positive outside. Okay, and if you want to just take a second to look at this picture, try to figure out what's going on. Figure out where the presynaptic and the postsynaptic neurons, which ones are the dendrites and which are the terminal ends. And if you go ahead and pause that, I'm about to answer that question for you. So this right here, whoopsie daisy, this right here is going to be the presynaptic neuron. And why do you know that? Well, you've got your neurotransmitters in the terminal end. So this becomes the terminal end of your presynaptic neuron, and this here is a dendrite. So a nerve impulse is going to be conducted that way, or possibly that way, but either way, going down from the dendrite here. And so the action potential has come up this branch and down to the terminal end. Take a minute here, see what you can label. Again, your presynaptic neuron is on top, postsynaptic's on the bottom. See if you can label everything labeled A through I. By the way, now would be a good time to pause so that when I start labeling stuff, I'm not getting you confused. Okay, so here's your synaptic transmission being broken down and well labeled. So you've got an action potential coming into the synaptic end, or the terminal end here. You've got mitochondria. And why do you need mitochondria? Because you are making tons and tons of energy in order to get exocytosis happening. And you also have the mitochondria because of the calcium pump that you've got there, which is pumping calcium out. Grab a pen here. Okay, pumping calcium out. So, sorry, try that again. Out. Here we go. So we're pumping calcium out. That's going to require an energy input, and we're pumping it out because anytime the signal comes all the way down to the synaptic cleft or to the terminal end here, the calcium is going to flood in, and we need a way of getting that back out so that a new action potential can arrive. So a vesicle here full of neurotransmitter, once calcium is pumped in, is going to flow out via exocytosis and release all of the products, in this case the neurotransmitter. Particularly, we want to make sure we know about acetylcholine, ACH. So that's going to flow into the synaptic cleft, which is then going to be picked up by one of these receptors right here. That is then going to tell the sodium channels to open up and allow a new action potential to head downstream. Now, if they were to just stay in the synaptic cleft and we've got like ACH just chilling here, then we're going to constantly be telling more sodium channels to open up, more flow inwards, and a constant action potential. So we need some way to break that down, right, and erase those. And that is going to be the job of acetylcholinesterase, which is then going to break down the ACH so that it can be reuptaken. I don't really know what the word is there, but reuptook into a new vesicle so that a new action potential can cause that new vesicle to fuse and the cycle can continue. Here's an example of an IB-like question, so you'd need to draw and label a diagram. Don't be surprised if you'd also need to annotate something like this. Again, showing the direction of nerve impulse propagation. That just means which direction is it going. So take a second, pause this, test yourself on it, and here we go. Okay, here's your cell body. Dendrites, your axon, terminal end, and terminal buttons. Oops, undo that last little bit here, sorry. Nucleus within the inside, and your nerve impulse propagation, your action potential, goes from the cell body down to the terminal end. Here's another IB question for you: how a nerve impulse passes along a non-myelinated, so we're thinking about something that doesn't have the myelin sheath, so just a typical nerve cell like we've been drawing, something like that, instead of the ones that have the myelin around it. So how does that move down? How does the nerve impulse move along? Take a second and do that, and I'll show you the mark scheme for that. And here's your mark scheme, so take a second if you want to pause it to see what's actually on there. Let me zoom in for you. two more for you. Here's one about the principles of synaptic transmission. Again, you can go ahead and pause. And here's your mark scheme. Notice that there's a diagram there. Feel free to draw that. I'll zoom into the actual answers now. Hopefully you're able to read everything. And here's one of my favorite questions. It's going to give you a chance to really review everything that we've learned this year in terms of membrane transport. So outline the use of 4 methods of membrane transport in nerves and synapses. You're probably shaking your head. Think about it for a sec. Hit pause. Okay, moving on to the endocrine system. We know that for an endocrine process to work, you have some kind of stimulus causing the hormone to be secreted from a gland directly into the blood. It travels through the blood to a target tissue. So those 3 pieces there, the gland, the blood, and the target tissue are the main parts of the endocrine system initiated by some kind of stimulus. And negative feedback is the process where once you hit a certain limit, your body basically turns on a different switch to get everything back in line. That whole idea of you've got tolerances. Here we go. And if you go too high, then your body's going to do something to bring that level lower. If you go too low, it'll do something to bring it higher. And the glands that you'll need to know are going to be the pituitary, the thyroid, the adrenal, the pancreas, ovary, and testes. Don't worry about the thymus or the pineal gland. Okay, so for you to be able to maintain homeostasis, you're going to use the systems that we've talked about, the nervous system, the endocrine system. As a review, when we talk about homeostasis, we mean keeping the internal environment within safe limits. So everything's going to have a tolerance. You don't need to know what the tolerances are, with the exception of body temperature and blood pH. So those 2 you do need to know. For the rest of them, you just need to know that we do keep osmoregulation in check. Right? You have to regulate your osmotic balance. That's all a matter of maintaining homeostasis. Your CO2 concentration, because that ties in with your pH. Your body temperature, you need to know that around 37 degrees plus or minus a degree, and how you do that as well. Blood glucose concentration, you need to know. Don't worry about the exact numbers. You need to know how it's done. We'll be talking about that here in a second. And that blood pH needs to be between roughly 7.4 plus or minus 0.05 units. So, and how we do that is controlled by the kidneys and breathing. You don't need to worry as much about buffering agents for now. So take a second, look over this slide. Okay, here's a quick list of everything that you need to know for the hormones that we're going to talk about this year. Insulin and glucagon, we'll talk about a little bit more in the rest of this video. Adrenaline is responsible for your sympathetic nervous system, that fight or flight. Testosterone, FSH, LH, estrogen, and progesterone, we're all going to be talking about. So put this in your back burner. You can write it down, but not something that you need to know immediately. HCG, again, and oxytocin, those are going to have to do with pregnancy. So we'll keep those in mind, but not something that you need to know for this test coming up soon. But for chapter 6.6, you will. So again, insulin, glucagon, adrenaline, and testosterone. Let's throw that in there. You should know for the test or quiz on Thursday. And now on to looking at how does homeostasis actually work. Well, we are using something called a negative feedback. Negative feedback means that you're constantly monitoring for your temperature or for whatever it is that you're looking at. And if everything's good, then you're okay. Yay, you're monitoring, checking, everything's good. If you're too low, then you do something to go the opposite direction. So if you're too cold, you do something to warm up and then remonitor. Still too cold, continue to heat up. Then if you're okay, just keep on monitoring. If you get too hot, now do something to cool down. And you've got this constant feedback mechanism of if you go out of equilibrium to get yourself back in or back into homeostasis, okay? And so the process of keeping your body within a certain temperature range is called thermoregulation, thermo referring to heat, which is why we've got hypothermia, hypo meaning low, thermia meaning temperature, hyperthermia meaning too hot, very high temperature. So hypothermia happens if you drop below a certain threshold. That becomes the point at which metabolism can no longer continue. Hyperthermia is heat stroke. and again can be deadly. It starts to fry your brain. Literally, it's denaturing the proteins in your body. So how do you keep your body from getting into hypo or hyperthermic conditions? Well, your hypothalamus is in your brainstem, and your hypothalamus is constantly being that monitor. It's checking to see if you're too hot, too cold, or just right. And everything is just right, then nothing needs to change. As soon as you get to be too cold, we've got a whole bunch of processes that are going to happen. Your arterioles constrict. That's called vasoconstriction. Vaso meaning your vascular system, constriction meaning getting tighter. No sweating, your hairs stand up, and you have muscle spasms known as shivering. And these two kind of go together. Both of them are going to produce heat or trapping in heat. All of this is leading to a warming of the blood, which is going to, again, be rechecked. If the hypothalamus sees that it's still too cold, then all of these processes continue until you get to the point that you're too hot. That's going to shut off the area of too cold, make that go away, and now you're in the condition of being too hot. Everything on the opposite end of the spectrum happens. Vasodilation, sweat glands open up, and then you have a lack of muscle spasms. Your muscles tend to relax just a little bit, and the hairs are lying flat until your blood cools down enough. And so this constant monitoring is happening to make sure that you don't get hypo or hyperthermia. And if we want to take a little bit more of a closer look at keeping your body temperature in check, we can take a look at the two organs most responsible for it, which are going to be the hypothalamus and the pituitary gland. So the hypothalamus is the one that's checking to see if your temperature is too high or too low. So the stimulus would be cold. That stimulus gets relayed to the hypothalamus, and the hypothalamus then says, oh, we're too hot or too cold. Now, here's an integration between the nervous system and the endocrine system, because the hypothalamus is going to send a nerve signal to the pituitary gland. The pituitary gland is then going to cause TSH to be released from the pituitary gland. That's thyroid stimulating hormone being released from the pituitary. That is a hormone that, as the name would suggest, is going to in turn cause an effect on the thyroid gland. That causes thyroid hormone to be released, which increases metabolism. Do you need to know this entire process? No. What you do need to understand is that we've got a connection between the nervous system and the endocrine system working to keep your body in check in homeostasis. And so when we look at the homeostasis of your blood glucose levels, I really like this diagram. I think it does a good job of taking everything that we've talked about and putting it all together. So when our blood glucose levels are high, here we go. When they're high, we're going to have the beta cells secreting insulin. Remember that once those blood sugar levels rise right after you eat, for example, insulin is secreted. That insulin is then going to have the target cell of the liver, and in the liver, glucose is going to be broken down into glycogen. Remember, glycogen is the long-term storage. Glucose is like your cash, right? So cash is going to go into savings. So high levels of money in your pocket, insulin is released to get some of that into savings. That way, if your blood sugar levels drop, glucagon, which is secreted by the alpha cells, can then take the money back out of savings, and glucose goes back into circulation. And so you've got this constant monitoring so that you get the right balance. Make sure you understand the alpha cells and the beta cells, where they're secreted from. Both of them are from the pancreas. What is being released, what is the conversion, and that this is constantly happening to maintain glucose levels at a very precise level. If you're not able to do that properly, then we call that diabetes. And you have two types of diabetes. The diagram's right here. Take a second to look that through. I think I'm at my time limit, so good luck on the quiz tomorrow. See you tomorrow.